Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss asexual reproduction and parasexual reproduction in fungi. First, I am going to tell about asexual reproduction. Asexual reproduction includes vegetative or somatic reproduction also, in which a new individual produced from unspecialized detached portion of the thallus. That means the plant body of fungi can give rise to a new individual, and that type of reproduction is called as vegetative reproduction or somatic reproduction. While in the case of asexual reproduction, a specialized reproductive bodies are produced from without involving nuclear fusion. So that process is called as asexual reproduction. Since there is no difference between asexual and vegetative reproduction, both being non-sexual, so they are put together under asexual reproduction. Then fungi can reproduce asexually by the following methods. They are fragmentation, budding, fission, chlamydospores, arthrospores or idea or thallospores, then finally sporangia and gonidia. First, I am going to tell about the fragmentation process. So, the fungal hyphae get broken into pieces, and each piece can produce into or emerge into a new fungi or established into a new colony of fungi, and that process is called as fragmentation. For example, yeast and molds. Then the next method, budding. Budding means a small soft portion of the cell wall get bulges out and the daughter nuclei migrated into it. So here you can find a small soft portion of the cell wall bulges out and the daughter nuclei migrated into the bulges area. Then this bulges area converted into a bud. Then the bud get detached from the mother cell by the process constriction. Then the bud are able to give rise to new fungi, and that process is called as budding. So example for budding fungi is yeast, that is Saccharomyces cerevisiae. Then the next method is called fission. So fission is the characteristic features of bacteria. So in fungi, only one fungi is reproduced by this method called fission, and that fungi is called as Schizosaccharomyces yeast or fission yeast. So fission means the fungi cell can divide into two equal daughter cells, and that process is called as binary fission or fission. Example, fission yeast or Schizosaccharomyces. Then the next method is called as chlamydospores. So the chlamydospores are thick-walled resting spores produced during unfavorable condition or adverse condition. So the chlamydospores are thick-walled resting spores. Then these structures are formed by thick wall around the cells. These chlamydospores are produced by thick wall around the cell. Then these chlamydospores are not detached from the hyphae. The chlamydospores are not detached from the hyphae. When the hyphae get detached, the other portion of the hyphae may die, but these chlamydospores are remain viable. During favorable condition, these chlamydospores can give rise to new fungi. For example, fusarium. Then the next method is called arthrospores or idea or thallospores. So the cells of the hyphae at the distal end, get round off and they get separated in basipetal succession. That means the separation starts from the apex towards the base of the hyphae. The separation starts from the apex and towards the base of the hyphae, and that succession is called as basiphytal succession. On germination, each thallospore give rise to new fungi. On germination, each thallospore may give rise to new fungi. The example for thallospore is Eripsae. Example for thallospore is Eripsae. Then sporangia and gonidia. The spores are produced internally, or the spores produced inside the sac-like structure called the sporangia or sporangium. The singular means sporangium, plural means sporangia. So a spore produced inside a sac-like structure is called as sporangia, and they are called as sporangiospores. And they are called as sporangiospores. So this sporangiospores may be flagellated or non-flagellated. The spore flagellated, if the spore is flagellated, it may be a uniflagellated or biflagellated, and that spores are called as zoospores. If the spores contains flagella, that spores are called as zoospores. Then if the spores not bears the flagella, and that spores are called as alpinospore. If the spores bears the flagella, that spores are called as zoospores. If the spores do not possess the flagella, that spores are called as alpinospores. So sporangia means the spores produced inside the sac-like structure called the sporangia or sporangium. So you can find the spores inside the sporangium. Then a stalk which bears the sporangium is called sporangiopore. While in the case of gonidia are produced externally. You can find these gonidias are produced externally. So gonidia means the spores produced externally. For example, penicillium. For sporangia, example is Rhizopus. For gonidia, example is penicillium. So that's all about the asexual reproduction in fungi. Then I am going to tell about the parasexual reproduction in fungi. Let me tell about parasexual reproduction in fungi. If the fungi lack sexual reproduction which involves nuclear fusion and meiosis, and that fungi is called as anamorphic fungi or fungi imperfecti fungi. So anamorphic fungi or imperfecti fungi lack sexual reproduction which involves karyogamy, that is nuclear fusion and meiosis. But the purpose of sexual reproduction is genetic recombination. So the genetic recombination is achieved by this fungi through a novel method called parasexual reproduction. So the genetic recombination is achieved by this fungi through a novel method called parasexual reproduction. So this parasexual reproduction is discovered by the scientist Pontocarva and Raper in the year 1952. In the fungi Aspergillus nudulans. So parasexual reproduction is discovered by the scientists Pontocarva and Raper in the year 1952 in the fungus Aspergillus nudulans. So that's all about the asexual reproduction and parasexual reproduction in fungi. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.